Gnana Deeksha, a concept-based digital learning. I'm Mohini Iyengar, lecturer in genetics from Telangana Social Welfare Residential Degree College for Women, Mahindra Hills. Today, we are going to learn about an interesting topic known as recombinant DNA technology. First, let's look at the learning outcomes. After today's class, we will have profound knowledge in tools and strategies used in recombinant DNA technology, and we will also understand the practical applications of recombinant DNA technology in various fields by studying some examples. Well, what is recombinant DNA? That's a very good question. Before we get into the details of the topic, I am sure most of you must be aware of the term DNA. DNA, it is the genetic material in most living organisms that stores all the information needed to create an organism. All DNA is made up of is a sugar, a phosphate group and one nitrogenous base. There are 4 nitrogenous bases, namely adenine, thymine, guanine and cytosine. The purines in the DNA are adenine and guanine. The pyrimidines in DNA are cytosine and thymine. So purines are larger than pyrimidines because they have a 2-membered ring structure, while pyrimidines just have single ring. In humans, the 4 nitrogenous bases adenine, thymine, guanine and cytosine are arranged as 3 billion base pairs, which essentially act as a blueprint for everything we do. The central dogma of molecular biology describes the flow of genetic information in cells from the DNA to the messenger RNA to the protein. What does it state? It states that genes specify the sequence of mRNA, and mRNA in turn will now code for protein. This was first proposed in 1958 by Francis Crick, the discoverer of the structure of DNA. For Crick, there were 4 kinds of information transfers. What were they? The first one was DNA to DNA transfer. It is called as DNA replication. The second was DNA to RNA information transfer. This is part of the first step of protein synthesis. Then RNA to protein is the second step of protein synthesis. And the final one is an RNA to RNA gene transfer, information transfer. This is seen in RNA viruses which copy themselves. Now, the process of DNA to mRNA information transfer is transcription, which is the first step in gene expression, where the DNA, like I said, is copied into an mRNA. Translation is the process by which the mRNA is decoded, translated to produce a polypeptide sequence, which is otherwise known as a protein. In a prokaryotic cell, transcription and translation occur simultaneously. They are coupled in nature, while in eukaryotes, the transcription occurs in the nucleus and the translation occurs in the cytoplasm. Now that we understand what DNA does, we should now understand an important point that the change in the DNA sequence can create a protein or inactivate a protein and alter the phenotype. What is a phenotype? Phenotype represents the observable characteristics or traits of an organism. Now, in ancient days, what humans do, they have looked at the phenotypes of the organisms and they have selectively bred or performed artificial selection for intentional reproduction of individuals in a population to have these desirable phenotypes. What they have done is they have selected the superior variety of animals or plants and produced hybrids or today's animals which are capable of functioning efficiently with desirable traits. With this in the background, I would like to ask you to carefully look at the slide for some examples of artificial selection or selective breeding. The first image shows a hybrid called mule. Mule is an offspring of a male donkey called Jack and a female horse called Med. Mules were created by Egyptians thousands of years ago as they were more patient, hardy and long-lived than horses and are more intelligent than donkeys. Mules have a capacity to function much better compared to horses and donkeys. Another interesting example of artificial selection or selective breeding is a modern day corn. The teosinte, ancestor of the corn, had short corn cobs and very few kernels per plant. But upon artificial selection or selective breeding, we are now able to have a modern corn that can be a larger ear and have more rows of kernels per plant. So, by artificial selection, what we are able to do? We are able to produce crops with higher yield, shorter harvest time, higher resistance to pests, diseases and better in taste in somewhat natural way. However, hybrids formed do not be true. Creating offspring with different traits thus causes traits that can be lost during evolution. Also, the process of artificial selection or selective breeding takes a very long time. Thus, to solve this problem, scientists have developed modern technologies to create recombinant DNA. This allows us to produce new or desirable phenotypes in a very short period of time. Genetic engineering is the technology used to make a recombinant DNA by combining two or more different sequences obtained from different organisms. Recombinant DNA technology is a way of pursuing the genetic engineering in order to generate products for human use. Did you know that the first recombinant DNA was generated by Paulberg, Boyer and Kohn in 1973? Let me give you an example. Look at the slide. You observe a cat that glows in the dark. How is this possible? We all know that the cat eyes glow in the dark because the retina reflects the light. But is it possible to have a glowing cat? When the genome of the cat and the DNA from the jellyfish that glows in the dark were combined, it produced a glow-in-the-dark cat. This is possible because, as we discussed earlier, DNA is made up of 4 nitrogenous bases: adenine, thymine, guanine and cytosine. So if you carefully look at the slide, you will observe that a cat, mouse and humans have a sequence of DNA which is similar if not identical. That means the language is same but the order of A, T, G, C is different. This sequence is basically genetic code which is said to be universal. That means we can combine DNA from different organisms to create recombinant DNA. With this background, now let's look at the steps involved in recombinant DNA. There are basically 5 steps in recombinant DNA technology. The first step is isolation of both DNA fragments containing gene sequence to be cloned and a vector DNA. The first step is to basically isolate your gene of interest and combine it with a vector molecule to be able to propagate into a host cell. Restriction digestion is the second step in which both the DNA strands that you have picked up, one the fragment of interest and 2 the vector, have to be cut with restriction endonucleases, preferably the same restriction endonuclease enzyme to create sticky ends or cohesive ends. Ligation of the sticky ends of the DNA fragments will be carried out by an enzyme called DNA ligase, that is the reaction called ligation. Ligation of the gene sequence of interest with the vector DNA will now form our recombinant DNA molecule. Once we have the recombinant DNA molecule ready, The next step is to transfer the recombinant DNA into an appropriate host. In this step, the recombinant DNA is introduced into a recipient host, mostly a bacterial cell. This process is called transformation. The last step, however, will be selection and multiplication of host cells carrying the recombinant DNA to get genetically identical population of cells called gene clone. Now, the transformation process basically generates a mixed population of cells where some of your cells which are carrying the recombinant DNA can be present along with some of the cells which do not have the recombinant DNA transformed. Now, the selection of the transformed host cells depends upon the type of the vector used and the marker that the vector carries. Now, why do you need to have a clone? Clone is a genetically identical copy of the gene. I will give you an example of why you need to have a clone. Let's imagine there is a company of a phone which manufactures a high-end phone with great features. Is it enough for the company to just manufacture one mobile phone and that's it? No, isn't it? So a company has to make a blueprint and manufacture many phones to allow the customers to buy the phone and get profit out of it. The same way, we perform gene cloning, a technique in recombinant DNA technology, to obtain our desired product in multiple copies. Now we understood the basic steps of recombinant DNA technology. But to understand a bit more deeper, we need to know the tools used in recombinant DNA technology. Just for our convenience, we have classified the tools of recombinant DNA technology into three types: enzymes, vectors, and hosts. Let's try to understand each of them in detail. Enzymes used in recombinant DNA technology basically are involved in editing or manipulating the DNA. So DNA manipulating enzymes are those that basically cut the DNA fragments at specific locations. Those enzymes that cut the DNA at specific locations are called restriction endonucleases. Arbor, Nathanson Smith won the Nobel Prize in 1978 for the discovery of restriction endonucleases enzymes. Now, at the advent of the discovery of these enzymes, today we are able to perform the recombinant DNA technology. These enzymes cut the DNA. What is the equipment that's used to cut? You use a scissors, right? So, molecular scissors are your restriction endonucleases that specifically cut the DNA at a unique sequence called recognition site or restriction site. Some of the examples of restriction endonucleases are EcoR1, BAMH1, HIND3. So, EcoR1 is the first restriction endonucleases that was isolated from bacteria, Escherichia coli. The second important enzyme in recombinant DNA technology are DNA ligases that act as molecular glue. Molecular glue, what does it do? It is responsible for the formation of phosphodiester bond between the two DNA fragments that form the recombinant DNA. So, ligation is brought about by DNA ligase. An example is a T4 DNA ligase. Apart from molecular scissors restriction endonuclease and molecular glue DNA ligase, there are other enzymes that are involved in recombinant DNA technology. They include polymerases. DNA polymerases are enzymes that catalyze the synthesis of a new strand of DNA from a pre-existing strand. There are different types of DNA polymerases that are used in recombinant DNA technology, namely E. coli DNA polymerase 1, a Clino fragment, thermostable Taq DNA polymerase, and a reverse transcriptase. Apart from that, DNA modifying enzymes are also involved in either addition or deletion of different chemical groups from DNA molecules. For example, alkaline phosphatases, which removes the phosphate group from the 5' terminus of the DNA. Polynucleotide kinases, these exactly play the opposite role of alkaline phosphatases, where they catalyze the transfer of phosphate group from ATP to the 5' terminus of DNA molecule. And the last important enzyme used in recombinant DNA technology are terminal transferases. This group of enzymes basically catalyze the addition of one or more deoxyribonucleotides to the 3' terminus of DNA molecules. We will now move on to the second important tools in recombinant DNA technology called vector molecules. Vectors are cloning vehicles that carry your gene of interest into a given host. If you remember, if you have to travel to a different place, what is the mode of transport? You prefer a shorter distance by a car, a very far distance by a ship or an airplane. Similarly, a vector is a molecule that actually transports your DNA of interest into a host for propagation. Now, vectors should have specific properties, especially having a new origin of replication, a selectable marker, a unique restriction site, and they should be very small in size for easy handling. Now, when you understand the features and criterion of what a vector should be, let's look at the type of vector depending upon the size of foreign DNA. Plasmids, cosmids, bacteriophage vectors and artificial chromosomes are some of the types of vectors that are used in recombinant DNA technology. Plasmids are double-stranded circular DNA molecules that autonomously replicate in bacteria which are present in many copies. They can carry and insert up to 5 to 10 kb in size. Some of the examples of the plasmid include PBR322 and POC vectors. Bacteriophage lambda has a linear viral DNA that can take up to 20 kilobases fragment. These bacteriophages are viruses that infect the bacteria and help in propagating your gene of interest. Cosmids are basically hybrids of a plasmid and a phage which can carry a double size of around 50 kb fragment into the host cell. Some of the examples include CRP2. And last but not the least, the artificial chromosomes, basically a yeast DNA containing a centromere, telomere and an origin of replication which can carry up to 1 million base pair in size. So yeast artificial chromosomes are the examples of artificial chromosomes. Moving on to the host cells. What are the hosts? Hosts are the living cells in which your recombinant DNA can be propagated. Mostly there are two important types of hosts used in recombinant DNA technology. Ones are prokaryotes like bacteria, specifically E. coli, eukaryotes like yeast, plant cells and animal cells. Now, host cell is basically depending upon the type of experiment that you've planned. If you want to express a prokaryotic gene, a prokaryotic host would do. But if you want a eukaryotic gene to be expressed, due to the limitation of complex machinery in prokaryotes, we need to use a eukaryotic host. So most commonly used eukaryotic hosts are yeast. Till now, we have learnt about cell-based gene cloning technique. However, if restriction sites are incompatible or if there is no restriction site available in and around the gene insert, then we can perform what is called as cell-free gene cloning. This is achieved by a technique called as polymerase chain reaction. Polymerase chain reaction is also called PCR. It was invented by Carey Mullis in 1983 and was patented in 1985. Its principle is based on the use of DNA polymerases as an in vitro replication of specific DNA sequences. There are three basic steps in polymerase chain reaction that include denaturation, annealing and extension. Denaturation is heat separation of denaturing DNA to obtain individual single-stranded template strands, for which the annealing of the synthesis synthesize a complementary sequence on the single-stranded template DNA. Once you have your primer annealing, then we extend it by polymerases or polymerization reaction using Taq DNA polymerase. PCR has vast amount of applications in performing cell-free cloning. They include the diagnosis, identification of pathogens using viral DNA or bacterial DNA, and it is also possible to amplify different regions of DNA obtained from a forensic scene, where a small amount of sample that is obtained from the crime scene can be used to amplify multiple copies of it for understanding the role of the criminal in the crime scene. I would like to summarize what we have learned until now. We have learned about recombinant DNA technology. What is the concept? What are the steps involved in recombinant DNA technology? And what are the tools used in recombinant DNA technology? Now, we know that we can join the DNA from two different organisms using recombinant DNA technology. Let's look at the applications in various fields of science and technology about recombinant DNA. Like I said, human life is basically affected by three important factors. 1. Good food or nutritious food. 2. Safe, healthy life. 3 is safe surroundings or environment. Recombinant DNA technology is playing an important role in improving health conditions by development of new drugs, vaccines, diagnostic kits, etc. Further, it has also helped us in developing transgenic organisms, where we can create a bacterium that's able to produce proteins which are of human use. Recombinant DNA technology also brought improvement not only in plants, animals, but also in microorganisms, which are used in making the environment clean by a process called bioremediation. Now, let's look at some of the examples in particular. In medicine, recombinant DNA technology is used for the production of humulin. Humulin is the first biosynthetic human insulin protein produced from E. coli, which is used to treat diabetes. Did you know that a normal human requires 100 units of insulin per day? Thanks to recombinant DNA technology, today, India being the diabetic capital of the world, we are able to meet the needs of diabetic population. Another good example of RDNA technology application in medicine is Hepatitis B virus, which is produced by using a surface antigen of the virus in stimulating the production of antibodies. It's a very safe technique that develops vaccines. The commercial product is called BVAC. All of you know, cancer is one of the most important health problems because many cases are difficult to prevent and treat. Monoclonal antibodies are the antibodies that have the specificity that bind specifically to cancer cells and kill them, are now developed using recombinant DNA technology. These monoclonal antibodies target specific cancer cells and are used in treatment of several malignancies like liver, prostate and other cancers. Now, most important application of recombinant DNA technology is DNA fingerprinting in forensic science. We all know that no two people have the same fingerprint. So, our fingerprints are very unique. Recombinant DNA technology, on the basis of restriction fragment length polymorphism, has developed DNA fingerprinting or DNA profiling that enables us to create a unique pattern of restriction sites such that once analyzed, each individual has a unique DNA fingerprint. This is helpful for paternity testing, or it is used in forensics for the identification of criminals. In the production of transgenic plants and animals, recombinant DNA technology has been used for production of golden rice and BT cotton flavor savored tomato. What are these? Golden rice is the variety of rice that carries a beta-carotene gene, which is the precursor of vitamin A that's used to treat the deficiency of vitamin A in the world. Similarly, BT cotton is a pest-resistant plant variety that's been produced by introducing a BT toxin obtained from Bacillus thuringiensis that kills the bollworm, which basically causes 50% of loss of cotton plants. Similarly, another variety of tomato called flavor saver tomato has been produced, which is silenced by a gene called polygalacturonase, which delays the fruit decay. We all know that the shelf life of tomato is shorter. So the transgenic variety is now able to survive for a longer periods of time at room temperature. Now let's look at some of the transgenic animals. What are transgenic animals? Like I said, those animals which contain the gene of interest obtained from another source introduced into it for the production of some of the pharmaceutically important products. Since we are using animals for production of pharmaceuticals, the technique is called molecular farming. Some of the examples are cow, which is used for the production of interferons that provide resistance to viruses in the milk. Similarly, sheep is used for the production of alpha-1 antitrypsin. Pig is used for the production of hemophilia factor 8, which is used for the treatment of hemophilia. Now you understood the uses for production of transgenic plants and animals. Let's now see how we are safeguarding the environment using recombinant DNA technology. We are able to produce biologically efficient fuels using genetic engineering. This is possible by modifying the microorganisms which are methanogens that produce biogas. Bioremediation, I mentioned earlier, is a technique of using microorganisms to break down the pollutants in the soil, air and water. So these microorganisms are capable of degrading pesticides, insecticides and fungicides. Another important application in safeguarding the environment is bioremediation. It is the use of organisms like microorganisms that genetically modified to break down pollutants in soil, air and groundwater. Degradation of pesticides, insecticides and fungicides in soil microbes like Pseudomonas has been used in recombinant DNA technology. Applications in industry. So apart from the application of recombinant DNA technology in developing transgenic organisms in medicine, we also have applications in industry. So genetically designed bacteria are put to use for generating industrial chemicals. A variety of organic chemicals can be synthesized at a large scale with the help of genetic engineering. Some of the examples include glucose. Glucose is synthesized from sucrose with the help of commercially used enzymes using genetically modified organisms, and cyanobacteria have been developed which can synthesize ammonia at a large scale that can be used as a fertilizer at a much cheaper cost. As you might have noticed, there are a great set of applications of recombinant DNA technology, but let us compare the pros and cons of this technology. We understood that it's possible to get a large quantity of product and any two organisms' genome can be recombined to get useful products. However, it might be harmful in developing byproducts which can destroy the organism's genome, and it's also leading to the loss of biodiversity as we are playing the gods and manipulating the genomes of the organisms. I hope that you understood what is the overview of recombinant DNA technology and what are its applications. Now, there is much to uncover in understanding the various applications of recombinant DNA technology. Before I conclude, I'd like to take a small assessment and understand whether you have understood the topic or not. So here are some of the questions for you. The enzyme that is used to cut the DNA is called as a restriction endonuclease, and the enzyme that is used to join two DNA fragments is called a DNA ligase. Autonomously replicating elements in the bacteria that carry the selectable markers are called plasmids. DNA fingerprinting is based on which of the following technologies? It's based on restriction fragment length polymorphism technology. It's also abbreviated as RFLP. The process of using microorganisms to degrade the pollutants is called as bioremediation. All of you are aware of coronavirus, which is causing our COVID condition. Now, we can use recombinant DNA technology in developing the vaccine for corona. So, it is because of recombinant DNA technology in science that has made the human life easier for years to come. That's all from my end today, but I have an activity for you students. Please list out a few genetically modified foods and also look at what type of vaccine is being developed for coronavirus. I would like to summarize some of the important points again that we've learned in today's class. First thing is, we have learned that DNA being the genetic material, it's now possible to combine genome of any two different organisms to produce or create a recombinant DNA which is not seen in nature. So it's on us to create what kind of desirable traits we want. We can select any two different hosts and create a recombinant DNA. Apart from that, we have learned that there are ample applications of recombinant DNA technology in cases of medicine, in agriculture, in production of transgenic plants, transgenic animals, and in safeguarding environment by bioremediation or biofuel production. With this, I would like to conclude the session today.